Assalamu alaikum. Welcome to this live stream. I'm delighted to be again with Dr. Paul Nelson from the Discovery Institute. Welcome, Paul, back to the channel.
It's always fun to be here.
So, Paul, this is part 4 of orphan genes and other taxonomically restricted genes. So we're covering this in quite some detail, and recently you were at a conference in which you were giving some of the newer insights that you have in regards to this. So let's just recap a little bit about what is the implication of this area for you.
Well, actually,
what is the
before I give you my outline, the implications are several, and they extend right across biology. The first is that in the second half of the 20th century, basically from the discovery of DNA, the molecular structure of DNA, up until the year 2000. Although things were beginning to change in the late 90s, biology had a badly distorted, or not, let's not say distorted, a badly abbreviated picture of the genetic diversity of life on planet Earth. And because of that, theory construction, the task of building theories about origins, building theories about biological function, and so forth, those theories were distorted in light of this very poor signal. And the introduction of whole genome sequencing really opened a window on the actual diversity, genetic diversity, present among living things. And that has caused repercussions that continue to ripple outwards today with respect, in particular, as I'll discuss in this presentation, to the hypothesis of universal common descent, or the theory of universal common descent. Yeah. I don't think that theory is going to recover from the shock that it received from these discoveries. In fact, at the end, today, I will talk about streams of thought within evolutionary theory itself. So these are going to be scientists who are not advocates of intelligent design, as I am. They're thoroughgoing evolutionary biologists. They are saying we have to rethink the geometry of life on this planet, moving away from a single tree picture in light of what we have learned about the genetics of living things. So it's the consequences are really radical in the original sense of that adjective, of going to the root of something. And I find it just endlessly exciting because the pace of discovery is accelerating.
Yeah.
These technologies have made DNA sequencing very rapid and inexpensive. In fact, here in the States, a high school student doing nanopore sequencing published a paper, beautiful paper, on the genome sequence of a species that no one else had looked at. And this was a high school kid. So we're in a whole new era.
Absolutely.
And some of the other implications I will spell out in my presentation today.
Brilliant. Thank you so much for that. So we're going to start part 4 now. I've just, if you can please go back to your first slide.
There it is.
There we go. Over to you.
All right. Awesome. And I have to tell you, I think this series is just going to keep going because we'll have part 5 next time, I hope, with more discussion. And who knows? It's like I said, it's an ongoing revolution. All right. Quick overview. We're going to look at the trends in discovery of orphans and TRGs, taxonomically restricted genes is what that acronym stands for. How abundant are they? Where do we find them? Then we'll look at the functional roles of orphans. And this is a progressive outline in the sense that 1 bears on 2, 2 bears on 3, which is orphans in the tree of life. And then they all bear on 4, which is implications for new theories. So that's where we'll end up. And I don't want to give the viewer the sense that I'm trying to hype orphans. They don't really need any help in stimulating excitement and thought because they're fascinating. But we really want to understand their significance at a deep level and where their discovery may be leading us in the future. So a quick refresher on terminology. There's little orphan Annie. There's a pun in the term orphan. Within molecular biology, ORF stands for an open reading frame. That's a sequence of DNA that exhibits the properties, such as having a start codon, followed by a long series of amino acid specifying codons, and no in-frame stop codons, which is diagnostic of a protein coding gene. And by the way, today I will be talking almost entirely about protein coding DNA sequences. There are other orphans, of course, but we won't discuss them. Other types of orphans, but we won't discuss those today. By the standard metrics used to define evolutionary relationships, such as in the widely used matching algorithm BLAST, orphans have no genetic kin. Probably today, they're known more frequently as taxonomically restricted genes or lineage-specific genes. A team in Poland working on bacterial orphans, in a recent paper put it this way: taxonomically restricted genes are genes that are present only in a particular taxonomic unit and have no traceable evolutionary history. These genes have no homology to genes from other organisms, and therefore they're fundamentally important for the study of the emergence of organismal unique traits. And we'll come back to that. It's one of the most fascinating aspects of orphans, to my mind. They go on. TRGs that are present at the species and genus level garner special interest because they seem to be related to the niches in which organisms find themselves. This group is looking, of course, only at bacteria, but it seems to be the case that orphans generally relate to taxon-specific, species or genus-specific functional roles depending on environment and other circumstances. So let's look at the abundance and discovery trend. As I said, the invention of this technology has revolutionized biology and has led to uncovering a genetic universe that's vastly larger than we understood really throughout most of the 20th century. So you can picture this as living things of all types supplying their genetic dictionaries to databases like the NCBI genomic database, which is one that I use frequently. And you've got a variety of forms here, from bacterial all the way up to cetaceans. There's a minke whale down here in the corner. And as the genomic databases grow, signals emerge from the data. Before we get to those signals, here's a figure showing the growth of these databases, where we have on the y-axis here a log scale and time along the x-axis. There are two lines here. One is gene sequences, that's the blue line. The red line are whole genome shotgun sequences, so these would be complete genomes. And you'll see a big jump here as this technology of sequencing became practical and the data began to pour in. But keep in mind the sort of point of this figure is the y-axis and the fact that it is a log scale. So we have an explosive growth. As the NCBI puts it on their webpage, from 1982 to the present, the number of bases in GenBank has doubled approximately every 18 months. So we are on a vast, riding, if you will, a vast wave of data that continues to grow. And the pattern to keep in mind as we look at the next few publications, and I'm sort of reporting from the front lines, if you will. This is a survey of very interesting recent publications, bringing the viewer up to date on what's happening in this area. Keep in mind these curves. Okay, they're called rarefaction curves, or they're also known as collector curves. And we have on the y-axis again, these would be types of data that we're organizing. And here on the x-axis, we have number of observations. And when you see a red line like this, what it tells you is that you have not yet saturated the space in terms of your sample. Because every time you have a new observation here along the horizontal axis, your type of data bumps up. In other words, this curve is continuing to increase. It's not flattening. When you get a horizontal line, the blue line, and it stays flat, then you can say with some assurance, we've saturated the space in terms of our sampling. We've pretty much covered the waterfront, so to speak, and we can be reasonably sure that we're not going to be surprised by something that we haven't yet seen. So the difference between these two curves is very important. What this red curve tells you is there's a lot more out there to be learned. And until that line begins to flatten, keep looking. Okay, so I would encourage the viewer to go get this manuscript. It hasn't yet been published to my knowledge in a journal, but it's available as a preprint at bioRxiv from a group in Germany. And they are using an algorithm called Diamond that is really state-of-the-art for sequence matching. In fact, I'm involved in a project where we are using BLAST-P, which is very much slower than Diamond. And I think we're going to have to swap out BLAST-P in our analytical pipeline and use the version of Diamond that this team is using, because it's really so much faster at processing data. In any case, what they did is they took really a comprehensive survey, or they pooled in a sort of what's available today, asking the question, what's available today in the databases. They got themselves a very large sample of gene and protein sequences, and they clustered them. So this is there are, of course, more details in the paper itself, but a quick summary is they used a 30% sequence identity cutoff. So in my little cartoon here, I have A and B, and these regions of turquoise, these turquoise rectangles, represent identities along the reading frames of these genes. The areas that are not colored the same would be divergent, but when you see 30% identity of this sort, where it's, at different regions along the reading frame, by standard biological reasoning, most investigators would say these genes are homologous, and therefore they would be clustered together. And A and B, and then if you find C that has 30% identity, these are going to form a cluster, and they would represent a group of orthologs, all right? By definition, orthologs are not orphans. So approximately 19 billion sequences went into their analysis. 32% of them were orthologs. In other words, they had more than one element. They had at least, well, you could say n+1 sequences were in that cluster, meaning, again, by definition, those are not orphans. 68% were singletons. Now, this is their noun for what I'm calling orphans or TRGs. Okay, so that's a short summary of what they did in the project. Now, let's look at their curves, which are incredibly interesting. So along the x-axis here, we have the number of genomic assemblies that they were working with. And this figure, by the way, is just for eukaryotes. 5,155 eukaryotic assemblies. They are examining different genomes. And then along the vertical axis, we have the number of clusters. Now, notice that this curve is not coming down. All right, it's a monotonically increasing trend. And in the caption to this figure, they say what this indicates is that there's a lot of diversity yet to be seen in the existing protein universe, because this line is just going off here at roughly a 45-degree angle. It's not flattening. So their expectation is, as more species are examined, more protein clusters are going to be found, and this trend will continue. Now note, these are not orphans. We'll get to that in the next slide. But even though they aren't orphans, these clusters here that you see on the y-axis, they're still interesting, because what it's again telling you, there's a lot of protein diversity out there yet to be discovered. So all by itself, laying aside orphans entirely for the moment, this is cool. This is interesting. Now, here is a figure from their paper that does show the Orphans Fraction. So over here on the left, these are all the clusters that have n plus, well, it would be 2 in this case, because the next group are pairs. So here, this group, this is at least 2 sequences here, 2 here that have been clustered. And then the singletons, the orphans, are over here. Now, this is fraction of the data set covered. So how much have they been able to cluster? Well, it comes out to about 92% right here, this point right here. So most of the data set fits into this portion of the curve, or under that portion of the curve. Over here, we have the orphans. So you go from 92% to 100% of the data set. And the total number here of unique sequences is approaching 2 billion. Now, why is that significant? Well, for all kinds of reasons. But the one that I'm going to explore when we come to the end, I'm sort of saving the best for the end, is if you think about what an orphan means, how it's defined. This has unbelievable, oh, not unbelievable, I don't want to pick the wrong adjective. It has remarkable consequences for the size of sequence space. Come back to that at the end. So even though most of the data fit here in terms of clusters that you can form using the homology criteria that they set forth, this is a lot of genetic uniqueness, and that should give you pause. That's something really to think about. Now, what's interesting, among other things, look at the shape of this curve, and this is for all the data in their sample. Okay, now let's look at the shape of the curve just for eukaryotes. Notice it's almost the same. That you come to this point right here. So we're talking now, we're not including bacteria, we're not including archaea, we're looking just at eukaryotes. You get almost the same curve, where the point where the singleton fraction begins is right about here at 93%. And then you have this large region of unique sequences, or orphans, 7%. For the sample as a whole, it's 6%. The very close match of these two statistics tells you, I think, something very important, that you are picking up a biologically robust signal, that these curves really can almost be superimposed on each other. I've highlighted here the relevant portion of the caption of this figure. And again, I encourage the viewer, this is one of the most exciting papers I've read in a long time. Go to the bioRxiv. I'll supply all the references to Subor as usual. So you know, underneath the YouTube video, you can get the citations. Go get this paper, read it, and think about it. It's got tremendous consequences. Okay, let me tell you a personal story. And I may have recounted this in a previous stream with Subor. Frankly, I can't remember. But it's relevant in this context. So let's jump back 18 years. Okay, I'm skinnier. I have more hair. I have almost none now, but I'm a much younger person. And lecturing at Dartmouth University on this topic. Well, on sort of intelligent design generally, but the theme of my talk that evening was the design implications of orphan genes. And it was near the height of the intelligent design controversy in the United States. So the room was packed, and there were Dartmouth biology faculty there, including sitting right up near the front, I think maybe even in the front row, if I remember correctly, a microbiologist from the Dartmouth faculty. And I show this figure. This is from a paper published in 2005 by a group in the UK, answering or trying to answer this same question that I've been exploring, namely, what's going to be the trend for discovery of orphans? So they looked at 122 microbial genomes, which was, a tiny fraction of what's available today. But they're saying, wow, we're finding all these orphans in these different genomes. What's going to be the trend of discovery for them going forward? So what they have here along the x-axis is the number of non-orphans, and along the y-axis, the vertical axis, the number of orphan genes. And they divided their data into two bins. The dark line here, the black boxes, these are shorter orphans, that is less than 150 amino acids. Here, the empty boxes, or the lighter boxes, are the longer orphans, longer than 150 amino acids, with an arbitrary cutoff in protein length of 150. But neither of these lines is coming down. And you can see when these species were sequenced, you had a big jump in the number of orphans right there. Okay, so I show this figure, and I'm already an enthusiast for orphans. I'm already getting very sort of evangelical about their significance, right, for biology. At that time, 2005, I think it was the fall of 2005. And the Q&A arrives, and this microbiologist is clearly upset. And you can tell, those of you who do public speaking, when you're standing up at the front of a room at the podium, looking out at all that sea of faces there, you can see who's enjoying the talk, who's not enjoying the talk, who's sleeping, whatever. I can see as I'm speaking that this guy wants to ask the first question. So the Q&A arrives, and he shoots his hand up, and he says to me, Paul, what's in the numerator position for that survey? And I say 122 genomes. And he says, what's in the denominator position? And I say, I don't know. He goes, nobody does, but it's bound to be this. Right? Orders of magnitude greater than 122 within the domains archaea and bacteria. And I don't know how many archaeal genomes were available at that time. It couldn't have been a very large number at that time, 18 years ago. He said, you have here a classic bad sample where you've taken a small portion, a fraction of the relevant data, and you're generalizing over the whole domain, and you're bound to be wrong. And he said, orphans are going to turn out to be statistical artifacts of a poor sample. In other words, as we sequence more and more species and get a better picture of genetic diversity, these apparently unique sequences are going to go away. And he was pretty vehement about this, right? So I say, well, yeah, 122 is not very big relative to probably millions of bacterial species. In any case, I would like to get back there someday. And if he's still on the faculty, I'm going to take him out to coffee or lunch and say, you know what? I was right. Because the signal did not go away. So this is, again, taken from an unpublished manuscript, and I'll, again, provide the citation to Subor. This is at bioRxiv also. A Polish group, I cited them earlier in the talk, looked at the trend in the domain bacteria. So here's an easier way to see their data. It looked at over 62,000 species. So 2 orders of magnitude plus some greater than what I was citing in 2005. Almost 200 million bacterial proteins. Orphans fraction, not quite 4.5%, a little bit less than 4.5%, divided into genus-specific orphans and species-specific orphans. Those together giving you 4.38%. Look at this. My guess is this is 100%. And what we see here is an error bar that there's probably some noise in the data, and this should really be 100%. I mean, that's my hunch, right? Maybe it's not, but I'm betting that it is. These curves are not flattening. Now, someday they will. Okay, so there's not an infinite number of bacterial and archaeal or bacterial cells on Earth. Someday they will, but they show no signs. These curves show no signs of flattening anytime soon. Okay, we're riding this red line off this way. Now, this is a central, I've just described over the past 10 minutes, it's a central motivating puzzle for researchers in the field. And I should tell you that orphan genes have attracted some of the brightest young talent in biology, which is exciting to me because it means that very smart people are trying very hard, to define what an orphan is, to get a precise handle on that concept, but B, to look at, trend of discovery, abundance, distribution, and C, finally, biological role. So these three authors said in a paper in 2020, this demands an explanation. The fact that every genome we look at has orphans, and even though our databases are growing enormously, NCBI doubling every 18 months, they're still present. We still find them, which means they're not a statistical artifact. All right, let's shift gears now. We've had a view, quick overview of abundance and discovery trend. Now let's look at functional roles. So let me take you back to I must be getting really old because old people like to reminisce. And I'm going to reminisce briefly about the way that I learned phylogenetics as a student prior to the genome era. So here is a phylogeny using cytochrome C, which is a rather small protein, 104 amino acids. And we see a branching pattern here relating these 7 mammalian species, their putative evolutionary history. And the little arrows here show changes in the cytochrome sequence along the branches of this phylogeny. Here's a representation of cytochrome itself. It's involved in electron transport and respiration in the membrane of mitochondria. Widely distributed protein, as you'll see in a moment. Okay, this is how I learned phylogenetics. Trees built on the basis of a single gene, a single protein, because those were the data that were available, right? We didn't have whole genomes. So here's an alignment showing, you can see across here the 104 amino acid sequence, the second half of it roughly. So we're starting here at 45 and going over here to 104. So we're looking at just a portion of the amino acid sequence. Here we have mammals. Here are other vertebrates, not mammals, birds, some reptiles there and so forth, some fish. Here we see these are arthropods, insects, yeast, and then a bunch of other groups, plants and so forth. And you'll notice just visually looking at this alignment, this is highly conserved. So down here across the bottom, and I apologize that I've never been able to get rid of this. I don't know how you do that. I'm technologically incompetent. So I apologize that this is on your screen. But you can see the counts of amino acid differences among these cytochrome C amino acid sequences very little variation in this molecule. I mean, there is some, right? Enough that you can get a putative evolutionary signal, but it's pretty much the same protein from one group to the next. Now, here's a little cartoon showing its role in the complex structure involved in electron transport during respiration. Here it is, hustling along, doing its job. Now, think about what's represented here. These are 7 mammalian species. Mammalian. We've got bats, rabbits, 3 types of primate. There's chimps, and we are primates. So here I am with Larry Moran of the University of Toronto. This is at the 2016 Royal Society meeting in London. All mammals, but notice the diversity of form here. A bat is not a chimp, and a chimp is not a human. So we have even though we're still within mammalia, we have a great deal of morphological and other kinds of disparity that's left untouched by cytochrome C. Now, at this point, you need to stop and think and say, if evolution is mainly a theory of transformation, where is the transformation explaining feature in cytochrome, in that 104 amino acid protein, that will account for these rather striking differences among mammals? Okay, a bat, like I said a moment ago, a bat is not a chimp. And if you're looking at what's effectively the same protein in those two groups, that's giving you effectively zero information about how bats came to be bats versus chimps and so forth. Fact is, when you go down this vertical axis here in the figure, mammals, other vertebrates, insects, yeast, variety of plants and such, this protein is really not telling you really not giving you any evolutionary, namely transformation explaining information at all. So if we go back here, we can say, well, let's put yeast in there and let's put tobacco in there because they have cytochrome C. And you can see that we are entirely at the wrong phenomenological level to understand how these different species came to be the way they are. Now, that should give you pause. And if it doesn't, consider this parallel. There is the P-51 fighter, a military aircraft widely used during the Second World War, roughly the second half of the Second World War, and then into the Korean War, an American fighter plane. Here is the F-35, the latest generation of American fighter planes. They're both airplanes, but this one can go a whole lot faster than this one. F-35s have amazing abilities to basically stop in place and hover. They have directional thrust that allows them to do really miraculous things in the air. So they're both airplanes, but they're very different. How are you going to explain the similarities and differences between these two types of military aircraft? Well, you're not going to do it in terms of this, a metal screw. So to extend the analogy or to illuminate it somewhat, cytochrome C is effectively a metal screw. It is entirely at the wrong phenomenological level to understand the evolutionary path from a series of common ancestors from a series of common ancestors to forms as diverse as a chimp or a bat or tobacco or yeast. Or you can find the same screw in a vacuum cleaner, the same screw that is in a vacuum cleaner or a chainsaw. Now, this is a problem because evolution, after all, is not a theory of similarity. It is a theory of transformation. And you simply cannot understand the transformation of form and function with respect to only a tiny fraction of the proteome, that is the total number of proteins in a species, or the genome, where you're looking at a little bit, a tiny little bit of the relevant data. So here, a typical mammalian or a typical animal proteome will be on the order of 20,000 proteins. This is 3.10 to the 9 base pairs, represents the human genome, and the total nucleic acid length for cytochrome C is 312. So you're looking at a tiny fraction of the relevant information. Not surprisingly, going from when I was a student, and I'm getting old, so this is a long time ago, going from that period to today, single gene and protein phylogenies are no longer constructed, in part because we have a lot more data, but also because investigators have realized that you need much more than a fraction to illuminate the process of evolution. It's a little cartoon to help make the point. There's cytochrome C, and these figures I'm going to show you are not to scale. I've made them sufficiently big so that you can see them. There's cytochrome C. And in the period when it was being employed to build phylogenies, roughly second half of the 20th century, pretty much the rest was unknown. So think of this rectangle as the genome of some species. And we've isolated cytochrome C laboriously, very laboriously, because sequencing proteins takes a lot of work, sequenced it, and we've got the sequence for cytochrome C for, let's say, an array of mammalian species. Along comes whole genome sequencing, and now we can look at the entire genome. And we have a great deal more information. And now these evolutionary trees are being constructed on the basis of very much larger pools of information, including whole genomes. But that has brought along with it a paradox, as you'll see in a moment. Because, let's start with tribolium. Here's the red flower beetle, a very well-studied insect. It's an agricultural pest. Tribolium appears to build its limbs with an orphan gene called flip-flop. So this German group looked at how the limbs extend from the trunk when they are developing. That's what this image shows here. These are the regions where flip-flop is active. If you want your legs as a red flower beetle to grow out from your body, which is kind of important to have a functional limb, you need flip-flop. Now, why is flip-flop interesting? Well, you can see in the title of the paper, it's an orphan. They could not find the corresponding sequences for flip-flop in other insect species. So it appears, at least at the moment, or at the time this was published, I was not able to check today, so I should probably do that. But in any case, when this was published, flip-flop was found only in tribolium. All right, if you think back to that figure, just go back to it. Going about flip-flop, we're in here. We're not over here, but it's in here in the total collection of relevant data that we have to explain the transformation, if it happens, of one kind of species into another. So what the take home from this kind of publication is, or what it tells you, is if you want to understand how tribolium came to be, as we find it today, you're going to have a story for where flip-flop came from, because it's demonstrably necessary for the normal formation of limbs. Okay, another example, an insect. A little leafhopper, looks coloration like an abstract expressionist painting. I just love the creativity of that carapace. This leafhopper spends its life, doing its thing under dew and rain and being exposed to water in a whole variety of circumstances, and it produces in its Malpighian tubules a super hydrophobic coating that it applies to its exterior. And when you look At that superhydrophobic coating, it's made up of proteins called brochosomins that form these little sort of buckyball-shaped structures. Remarkable, okay? Why is this interesting? It's interesting because the proteins involved in making those brochosomes. Brochosomins are the name of the proteins. Brochosomes are these little structures. Look how geometrically similar they are. It's really amazing. They're orphans. Now, why would the insect go to the trouble of making this coating? Well, probably because it needs it for its normal life. Its normal life history involves protecting itself from water. But when you look at the proteins, they seem to be, again, at the time of the publication, I really should go and make sure that that's still the case and go and check it. In fact, I'll try to do that before I send the references to Subor. In any case, at the time of this publication, these were orphans. Salamanders. Salamanders can regenerate their limbs. To my knowledge, a capability that is unique to that group. An orphan gene is necessary for limb regeneration and development in salamanders. Again, we're talking about not a peripheral aspect of the biology of these creatures. Having a limb and being able to regenerate a limb is a central requirement for a vertebrate, a tetrapod like a salamander. And when these investigators went looking for the homologs or the orthologs of this particular gene, they couldn't find them. All right. No ortholog is present in other groups. Apparently, it's a salamander-specific, taxonomically restricted gene. Last example, and we could do this all afternoon or evening. Other examples of the functional roles of orphans, they are not peripheral. They're central to the biology of their respective groups. So Hydra is a polyp, freshwater polyp, that grows in the water column. The embryos grow in the water column. I mean, they spend their whole life there, frankly. And what else is in the water column? Well, a lot of bacteria looking for a meal. So as Hydra are developing, the mother supplies antimicrobial peptides. This is from the PNAS publication in that journal. She supplies, if you will, gosh, I wish I could think of a good term. The medication, the therapy to keep the bacterial populations down so the embryo is not destroyed or parasitized by bacteria. Very important in that particular environment. Here you can see this has been lit up by a probe, a purple probe. Again, taxon specific. These investigators could not find these genes outside the genus Hydra. They're taxonomically restricted. So these are 5, I think that's the counts right, 5 examples of very important roles for orphans that would not have been revealed to us, if you will, if we didn't have the ability to look at the whole genome and see what's there. Last example, the most striking, I think, and the one that will take us into the tree of life part of the talk. So here is the classic 3-domain phylogeny of life on Earth, largely due to the work of the late Carl Woese. And we are here on this branch. We're an animal, so we sit over here in the eukaryotes. According to this picture, this geometry, there was a last eukaryotic common ancestor, Lika, not Luca, Lika, somewhere here, from which all other eukaryotes stem. What is a feature of eukaryotes with respect to their genetic information that defines our domain? Well, it is chromosomal segregation in 2 forms, mitosis and meiosis. So this cartoon shows mitosis, which is a cycle, cell division, chromosomal segregation, where the chromosomes are grabbed by specialized machinery and pulled apart towards the poles of a dividing cell. And this goes on all the time. Right now, as you're listening to me, you've got billions and billions of cells undergoing mitosis within your body. For instance, in your bone marrow, you're producing new cells all the time, and mitotic division is happening. So you can see here the spindle fiber apparatus separating chromosomes, which have paired along the midline. They're going to be pulled towards the poles. Familiar to you, I think, from even high school biology. And meiosis, which is not a cycle. It is chromosomal reduction as gametes are being produced in sexual reproduction. Okay, so you can see the number is going from diploid to haploid as these chromosomes are again being segregated, but you have cell division where the chromosome, it's like in Homo sapiens, we go from 46 to 23 in this process as we produce eggs and sperm. Very important to being a eukaryote is being able to do this. So it is, again, this is not peripheral. These are not peripheral features of the domain eukarya. Now, a group at the University of Toronto in this paper looked at the roles that, how these roles were being played in different, very well-studied groups, such as C. elegans, the nematode, and Saccharomyces yeast. And what they found was remarkable, that when you look at the protein machinery involved in chromosomal segregation in these groups, it appears to be not universal, but in fact lineage specific. So this is a quote from, I believe, the conclusions of their paper. Essential lineage specific taxonomically restricted genes in yeast and worms play key roles in chromosomal segregation. And they ask the question that you should ask: why should so many taxonomically restricted genes play key roles in this core process? Well, they don't say this. That is the rectangle in yellow here on the slide. But my tentative answer to that kind of question is, maybe we don't have life as n equals 1. In other words, that if universal common descent were the case, we have one sample of life in the whole universe. It's here on Earth as far as we know. And it's all the same basic operating systems with some filigrees here and there, right? That LUCA existed, and the essential hardware that we have in our cells, we inherited from LUCA going all the way back 3.5, 3.8 billion years, whenever life began. Counterpose that picture to what these data seem to be indicating. Or in this case, we're not going actually to LUCA, we're going to the last eukaryotic common ancestor, but the overall moral of the signal is the same. And I'll show you in a moment data that are relevant to LUCA. But the picture is not one operating system, multiple operating systems where the key features are not shared. All right, let's segue now to the tree of life, and I'm right on schedule. I'm very happy about that. Okay, these kinds of findings should be alerting you that there may be something wrong with that single tree picture. And so let's do that. Let's move into that area, beginning now to the third point in my outline, orphans in the tree of life. Okay, let's jump back 21 years to an early review article on whole genome, the whole genome revolution, from the late Russell Doolittle. I guess he died in 2019. Someone told me this past weekend. He was a member of the National Academy of Sciences at University of California, San Diego. And much of his career was devoted to looking at protein relationships. He specialized on the origin of the blood clotting system. But as a more general interest, he was very curious about these tree of life patterns and where proteins came from and how they were related. And in this review paper, again, 2002, there's not a lot of information available relative to what we have today, 21 years later. But even then, with just a handful of whole genomes available to look at, he picked up the signal. Vast numbers of putative genes for proteins of unknown function. Now keep that in mind. Why is unknown function relevant? He says, this is the biggest surprise. In every genome examined so far, at least a quarter of the genes remain hypothetical and that no function can be ascribed. He goes on. How could there be so much in the way of unknown equipment? There are large numbers of unidentified genes. Oh, there's a typo here. I've got to fix that. In a variety of organisms that look conventional in every way, where these unique sequences are coming from and what they do remain baffling mysteries. Now, why is that a mystery? It's a mystery because in his mind, as a unquestioned background, he has this picture, what I'm calling here the single tree scenario for the origin of the DNA, RNA, protein system that's ubiquitous across life on Earth. This happens once. It is captured in Luca, our ultimate grandmother, and therefore everything that we observe today, no matter what species we examine, will trace its history ultimately, 3.5 billion, whatever the date of Luca happens to be, back to that single theater scenario. To be more precise, when life got started, it wasn't Luca. It was the first universal common ancestor, but there's a continuous lineage from the first universal common ancestor to Luca, the last universal common ancestor. And then we have the branching that we see in this cartoon. Okay, Doolittle takes this as a given. It's part of what I call the furniture of reality, right? Like the Earth goes around the sun. The elements represent discrete kinds of atoms, that we see in the periodic table and so forth. In other words, the pieces of empirical knowledge that we have that no one thinks are up for grabs. So for Doolittle, this is not at issue. It's the case, right? So if you have this geometry in your mind, or here's another way of looking at it. Here we have the prebiotic world, pathways, chemical pathways leading to the living state. There is a dramatic bottleneck at Luca because none of these other lineages, if they existed, survived. This, again, is our ultimate grandmother. All diversity traces back to that singularity. So if you have this picture as a given, you are not going to expect to find a lot of unique genes and proteins. Now, let's come towards the present. It didn't take long, once whole genomes were available, for investigators to say, we have different methods, experimental methods of perturbing whole genomes to see what the essential hardware happens to be. So this is a cartoon showing a transposable DNA element called the transposon, hence the name. It moves around, that integrates itself randomly into other DNA. So here's a circular bacterial chromosome. We get 4 transposon integrations. When it integrates, it disrupts the reading frame, the gene in the native DNA, and at least one of those genes was essential. And you can't grow these bacteria. It's over, right? Your plates will be sterile. Same thing here. One of those integration sites hit essential hardware. In this case, however, transposons integrated, but they didn't land on genes that the cell couldn't do without, or the protein products that the cell couldn't do without. Well, if you do this often enough, repeat the process, you will be able to work your way right around this chromosome, especially if it's a fairly small chromosome. And the very fact that you see in live cells transposon integration sites tells you that wherever this landed is probably not essential under lab conditions. So it's a remarkable kind of experiment. The name is transposed on immunogenesis. These are knockouts, so we've eliminated the genetic information at these sites. The cell's alive, and we can sequence the DNA, map the transposon integration sites, and figure out what does the cell need to survive. So this experiment has been done many times, and every time it's done, the results are interesting. So in Lucy Shapiro's lab at Stanford, they did this kind of experiment with Colobacter, which is a bacterial species that you can culture out of fresh water. And they found a little over 1,000 essential genome features, including 90 segments of unknown function. Now, that's nearly 1/10 of the essential hardware. Remember, I told you, keep your eye on this signal, unknown function, because here's how Shapiro, before I get to that, Shapiro herself was surprised. We found these essential DNA segments. We don't know what they're doing. Why is that relevant to orphan status? Well, think about how genome data are deposited in databases. They're not just dumped in there. They're annotated with respect to their functional roles. So in this cartoon, here is our database. And these sequences, DNA sequences, have been deposited there, and they have associated functional roles. So we align. This is on the top. This is our new sequence. And it turns out it matches with a ribosomal protein or a gene that produces a ribosomal protein. Now we know what this gene is doing, and this sequence can be added to the database. And that's how the genetic dictionary grows. Except with orphans, there is no associated function because there's no associated match. Using BLAST or some other matching algorithm, we find no hits in the database, telling us because there's no function, it's probably an orphan, at least when we first sequence it and submit it for matching. So that's why unknown function is a strong clue that you may be dealing with an orphan. So here's a figure from Shapiro's paper that every time I look at it, kind of, I wouldn't say it gives me chills. That would be overstating the point. But I find it really, really interesting because Colobacter and E. coli are proteobacteria. They're in that bacterial clade. They're putative evolutionary kin. But notice that Colobacter is running its cell biology using unique sequences and proteins and other products from its genome that are essential for its existence that are not shared with E. coli and vice versa. Now, if you did this same experiment with another member of the proteobacteria, chances are you're going to see very much the same Venn diagram. A lot of hardware that's shared and some that's taxon specific. This is very significant. It turns out, and I'm getting close to the end now, still on schedule, it turns out that across the tree of life, very few of the essential functions of organisms are using the same molecular actors across all three domains. So here is the classic three-domain picture where we have LUCA here at the origin, archaea, bacteria, eukarya. Now, if LUCA sits here, your expectation would be that as it leaves offspring, and we come from LUCA towards the present, that the essential features of LUCA, which would have had a DNA genome, would have had ribosomes, it would have had DNA replication and transcription, machinery like polymerases, it would have had a lot of molecular hardware that is demonstrably essential for the living state. So the expectation from this sort of geometry is the essential hardware is going to be conserved as one moves out into the three domains. All right, it's textbook prediction. It turned out that it's not the case. So here was here is an experiment done with an archaean, Sulfolobus. Excuse me. It is a single-celled organism that lives in what's called a hyperthermophilic environment, very, very high temperatures, sulfur metabolizing. And I realize this is kind of a busy figure, so let me explain to you what's going on here. Along this axis, we have different groups. So the first two are archaea, all right, Sulfolobus and Methanococcus. The next two are eukaryotes. These are both yeast. Then we have three bacterial groups, Bacillus, E. coli, Bacteroides. So we have representatives of all three major domains here along this axis, and we have Mycoplasma Syn 3.0, which is Craig Venter's synthetic cell. Now, across the horizontal axis, we have representations of essential hardware. So we have three major classes: information storage and processing, metabolism, and cell processes, divided into their sub-processes. Now, these single letter codes here correspond to particular classes of function. Let me just jump over here. These single letter codes are found in the clusters of orthologous groups database, the COGS database. So you can see here we have translation. These are the genes and proteins involved in building the ribosome, RNA processing, cell cycle, and so forth. So that's what these single letter codes represent down here. So you see we have three codes here that correspond to the more general class of information storage and processing, several different codes here for metabolism and so forth. Now, this, it's kind of, what would you call that? Mauve? That's a salmon color. Anyway, this line, the salmon colored line running across this way, that's the genome of Sulfolobus that this experiment determined to be essential. Remember the title here, the essential genome. You can't have Sulfolobus if you don't have these genes and their products present. What's this? Poorly characterized. Over here, what is this category? Notice that there's nothing corresponding to these genes in these other groups. That's because these are probably Sulfolobus specific genes and proteins, right? And notice how patchy these distributions are. Where as you move across the domains, you'll find big white space because those genes are simply not present. Poorly characterized, though, should really get your attention. Why poorly characterized? Because the functions are unknown. And remember what I told you a moment ago about unknown function in relation to orphan status. The reason they're unknown function is there at the time of the publication of this paper, nothing in the databases corresponded to these sequences. Yet we know experimentally they are necessary for Sulfolobus to exist at all. Okay, it's a busy figure, but I called your attention to two important things. This very patchy distribution of essential cell hardware. Really, the place where it's most uniform is here. And I would bet, I don't know what I should bet. I don't have a great deal of money, so maybe I'll bet my personal library that these regions are ribosomal. Ribosome is that's what it means to be a cell on this planet, is to have a ribosome. So these are going to be very highly conserved. But as you go out this way, you find a very patchy pattern. And right over here, nothing. Nothing. Yet we know these cells need these products, or these genes in their products, to exist at all. And many of them are orphans, probably. Now, there's a term in the literature for this pattern. It's called non-orthologous gene displacement. Kind of, I'll sell you that term as a sleeping aid, all right? NOGD for short. But Eugene Koonin, the NCBI, the National Academy, looks at this and he says this pattern, where key hardware is not shared, extends across most of the functional systems and pathways in terrestrial life. As a consequence, the universal core of life has shrunk almost to the point of vanishing. With the exception of those in, proteins, genes and proteins involved in key steps in information transmission, there is no universal genetic core owing to the near ubiquity of non-orthologous gene displacement. Well, what are the consequences of these kinds of findings for Luca? For Doolittle, the basis of this kind of signal and others says Luca is a profoundly problematic entity. It didn't exist. There was no single common ancestral cell that gave rise to all the genomes we see today. One of the reasons for that conclusion, held by Doolittle, he says, is it would require an enormous genome, what he calls a genome of Eden. And that hypothesis, he says, with some Canadian understatement, is unappealing. 2010, so that's 13 years ago now, he said Darwin arguably was wrong. And this emphasis, by the way, is in the original. That's Doolittle's italics. For most of life and most of its history, descent with modification is not the simple branching process Darwin envisioned. So this skepticism about the existence of Luca and universal common descent has been carried on by his collaborators, such as Eric Baptiste, who I believe was a postdoc with Doolittle about 20 years ago. But Baptiste, if anything, has gone even further than Doolittle. And in this paper, in 2021, he and his co-authors said, look, a glaring proportion of gene families is simply not shared among most cellular lineages. How and why is life massively reinventing its genes? We need to understand, these guys argue, that there are processes that give rise to genes that don't have homologs outside that particular lineage. And we need models of gene origination, they say, other than diverging from a single last common ancestor. So the next two figures I'm going to show you are rather difficult to see, and I need to carefully explain them. But they're very important. So again, remember these functional categories. These are sort of the major functional classes in any cell on the planet, including you. I mean, you're made up of trillions of cells, but your cells do these things. But here's the figure from their paper. And I realize it's a little hard to follow, so let me explain what's going on here. Those functional classes that I showed you on this slide are arrayed here along this axis of this rectangle. So starting at the top, we have transcription, cell cycle. The reason, by the way, that these two lines are blank, this section of the figure is empty, is these are eukaryotic relevant features. Let's see here. Yes, these involve nuclear proteins involved in the nucleus, and bacteria and archaea don't have nuclei. So that's why this section of the figure is empty. In any case, along this axis, we have these major functional classes. And the heat map here is how much of this is the dark portion is taxon specific and how much of it is universal, the light portion here. And you can see this is crazy patchwork. Here's an evolutionary tree for these groups, and they are not sharing key hardware relevant to these life-defining functional roles that they have to perform. It's a very patchy distribution. Another way of visualizing the same data is this figure, also from the paper by Watson and Baptiste and Lopez. All right, so again, we have a heat map here. And what this shows you is that the shared genes, what they call non-lineage specific, are relatively light colored along this spectrum here, from the darkest portion to the lightest portion. In other words, they're not as common or universal as one would expect, and the lineage specific, so we're talking about this line right here versus this line. The lineage specific genes are much farther along in this heat map. This is a much darker line than this one. The take home, these different groups from the archaea and the bacteria have very different ways of running their cell systems. They're not using the same genes. Okay, this is what you see here on this slide is not what one would expect from universal common descent or the existence of LUCA. These patterns of data strongly contradict that geometry. As a consequence, Doolittle and Baptiste did this. Goodbye, LUCA. And I believe Ford Doolittle drew this figure himself. Notice what's missing. There's no universal common ancestor down here. So this is the very early history of life, is represented by Ford Doolittle. You get this wild mangrove type pattern of interactions between the three domains, but there's no last universal common ancestor here. It's gone. Well, you can think of this as the theoretical biology equivalent of storming the Bastille, because the implications extend everywhere within evolutionary theory. And I'll tell you, if you think about this carefully and reflect on it, once you get rid of LUCA, at the base of the tree of life, the fracturing begins to extend upwards into the branches, and it becomes impossible to stop. I won't say more about that today. That's for another talk with Subor, maybe 2 or 3. But the fracturing becomes impossible to stop, and the tree of life ceases to be a single tree and begins to come apart at its base, and the fracturing process climbs up into the branches. All right. So here's Baptiste over here, and this is a screen capture from one of his lectures. He says, the notion of a tree of life is being increasingly questioned. And so he's reproducing the figure here from his mentor, Ford Doolittle. The simple notion of a single tree of life is gone forever. He's quoting Uri Wolf, who is one of Eugene Koonen's collaborators. Again, from the same lecture, another screen capture. The question, Baptiste says, should be, is there a tree of life? Not, how does the tree of life look? And he says, this is, this tree of life geometry has been somewhat misleading. So remember, Eric Baptiste is not an intelligent design advocate. He is a thoroughgoing evolutionary theorist. So he is being moved to this skepticism, not by any sort of metaphysical predisposition, favoring teleology, or God knows what negative thing you want to attach to intelligent design. This is being driven by his attempt to grapple with the data. He's hardly alone. Adam Rechlitz and Jeff Lawrence in this paper, again, this is now 13 years ago, so this controversy has been brewing for a long time. The title of the paper is The Inevitable Failure of the Tree of Life. I think that's the title of the paper. Okay, bracket that. Let me go and retrieve the reference. That may be the title of a section of their paper. All right. And I don't want to misrepresent them. In any case, they say there never was an established ancestral population. It didn't exist. Okay. This actually is something that I did not have time to explore today, about the way that universal common descent generates predictions by being coupled to other parts of our biological knowledge. But what these guys are doing in this paper is they're saying, we're going to hang on to the biological knowledge we have that's independent of universal common descent, and we're going to make universal common descent pay for the consequences of its failed predictions. All right, implications for new theory. Oh, shoot. I got to make this fast because I've run over the hour. If you think about how orphans are defined as having no detectable homology to other sequences, and project that into sequence space in its entirety. Now, sequence space is a high-dimensional abstract concept. It's a theoretical concept that cannot accurately be represented in a 2D Euclidean plane. So I apologize for this figure, But after all, it's a PowerPoint slide, right? I've got to do something. So I've compressed a very high dimensional space down to a two-dimensional plane. But what I want you to take home from this figure is the fact that an orphan, by definition, is at least equidistant from any other orphan or any other gene. At least equidistant. It may be very much farther away. But the spheres here are given to you by radii that extend. Each of these is an orphan. Each of these gray dots is an orphan. From each of them, using the bioinformatics criteria that define orphans, and a radius extends so that as you move out from known protein clusters and families into the orphans portion of sequence space, you have to travel a very long way indeed until you find the next orphan. And if they're in July 2023 are on current databases contain almost 2 billion orphans. Singletons is what that German group called them. We have, on an analogy to astronomy, we have an enormous galaxy or multiple galaxies extending way out into genetic space. Now, if you think about this with respect to the last universal common ancestor, it becomes more or less immediately impossible for a single cell to have given rise to the genetic diversity that we know today, that is going to continue to grow. And I would say this, even if I weren't an intelligent design advocate, if I can transform myself empathetically or imaginatively into an evolutionary biologist who's an atheist, all right? So I'll go to a wholly different part of theoretical space, and I would still come up with this finding. Namely, there's way too much genetic diversity to be fit into a single tree geometry. And it's got to be different. Of necessity given to us by cell biology, even the simplest cell that we know requires molecular diversity for function. Last slide. Let's suppose that Luca existed. Postulate that Luca existed. Well, we can extend a radius from Luca that's defined by three things. The functional demands of being alive. There's no dying in evolution. And you've got to be able to leave offspring. Known mutational processes and the constraints on protein variation. And time. That gives us a radius, and that gives us a sphere, outside of which we should not expect to observe genes and proteins. Again, assuming the existence of Luca. We're not going to find them outside that sphere. There hasn't been enough time. The mutational processes necessary will destroy the cell, and there's no dying in evolution, and you've got to be able to leave offspring. So, assuming that you can get good constraints or characterize these parameters in a rigorous way, A, B, and C, you can define a sphere, and I'm using a sphere simply as a convenience for the sake of this illustration. You can define a space, let's call it that, outside of which you should not expect to observe genes and proteins. Now, what do? How do you answer that question? You just sequence DNA, and you ask, where is it landing? Is it in here, in this sphere, or outside? And here again is where we pass from naturalistic evolutionary theory into the possibility of design. Because you let the organisms tell you where their genes and proteins are landing. Are they landing in here? There's that tiny little space defined by Luca, or are they somewhere out here? And this will happen in our lifetimes. We will be able to establish in that space, it's our genes and proteins in that space, or are they in this space? All right. I apologize for going on so long. Something, I really wish I could change about myself that I could finish on time. But in any case, I got the key points on the table. So
Brilliant. Thank you so much for that lovely, lucid presentation. I've actually got some questions based upon your presentation, but I thought I could postpone this for the next session because I wanted to actually ask you, since we're running out of time, about the recent discussion between Dennis Noble and Professor Richard Dawkins. So as somebody, I mean, I'm just going to ask you a question from, imagine it's the summer of 1998, you just graduated with your PhD from the University of Chicago, and someone tells you, well, roughly 25 something years from now, you're going to have a seismic shift, the selfish gene, the central dogma, Weismann's barrier, all this stuff is going to get challenged by systems biology. Would you have believed that this sort of discussion would take place?
No.
Okay, there you go.
No. I would have said, well, that's a pleasant fantasy. But I vividly remember Dawkins coming to give a talk at Chicago when I was a student. I think I was probably second or third year. And the room was packed. Probably we were violating the fire regulations for that particular auditorium. Every single seat was taken. People were standing in the back and along the walls, right? Almost anybody on that campus with any interest in evolutionary theory was jammed into that room. That was the authority and the prestige that Dawkins brought to campus that day. And I was sitting down front. I managed to get there early and get a decent seat. So I was actually not that far from Dawkins himself. And I remember, this is not an overstatement, he was kind of vibrating like a tuning fork. His intensity of focus, this is before he had the unhappy health problems that he suffered in recent years. Just tremendous presence and clarity. And the questions that were being fired at him by different evolutionary biologists on the Chicago faculty, he would just brush them aside like he was batting a shuttlecock in badminton, right? Such was his authority. And that genes first, bottom-up reductionist picture of biology where all biological change flows from mutations in nucleic acid outward to the phenotype was unquestioned. So as I remember, the questions that were being directed to him had to do with nuances in that picture.
Yeah.
What Dennis Noble has done and the systems biologists have done is they've stood that picture on its head. And Noble, in a series of really beautiful papers, has said there is no privileged level in any organism.
Yeah.
Yes, DNA is there. Yes, DNA is important. But Noble does this as a joke. It's really funny. He goes, I'll tell you what, you isolate some DNA from yeast, right? Or pick your organism, isolate the DNA, put it in a little flask, and leave it there.
Yeah.
Come back in 20 years, nothing will happen. DNA is a wonderful polymer. It can pack in a lot of information, but as far as a living thing is concerned, it's just completely sterile.
Yeah.
He said that fact alone, that DNA is chemically inert, should tell you that organisms are more than their DNA. Now, of course, Dawkins would acknowledge that, but he would say, nonetheless, and I think Noble was one of Dawkins' teachers at Oxford, if I'm not mistaken, so they've known each other for a long time. He would say, nonetheless, Professor Noble, my claim holds. Evolutionary change flows from nucleic acid outward. And Noble would say, I'm sorry, the evidence today indicates that that is simply not the case. So this is a debate within evolutionary theory that your thought experiment a moment ago, I never would have expected to see.
Yeah.
You know, and to me, it's indicative of a deep sea change within evolutionary theory that we are still going to see more and more of. The number of senior investigators who have worked up the courage to say, you know what, I don't buy that selfish gene picture anymore.
Yeah.
Is growing all the time. You know some of these people at Exeter, right? You've interacted with them. Yeah. it reminds me a little bit of what happens in a political revolution, where you get a sudden flourishing of alternative positions as people sense that the authority of the central control political party, let's say, has been diminished. And then all of a sudden people are saying, well, you know what? I never believed it to begin with, but to protect myself, to get funding, to have a career, I more or less went with everybody else. But since I see he doesn't believe it and she doesn't believe it, right? I'm going to stand up now and say, you know what? I don't believe it either. I've always had problems with that selfish gene picture. And you get this flourishing of alternative positions. Yeah. Dawkins, I think, very late in his career now, very late, has come to see that things have gone and run away. So he's been very involved in these debates, for instance, in New Zealand about folk knowledge having an equal place with empirical science and
irrelevant things. Yeah. So I want to ask you a burning question here, which came to my mind, and I'm sure is coming to the mind of a lot of thinkers at the moment. What are the design implications for this paradigm shift? If we are to, we are going to sort of go out on a limb, right? Because it's early days. But what would you say are some of the design implications of a seismic shift towards systems biology from the central dogma perspective?
Well, the first one that leaps to mind is reductionism fails.
Oh, yeah.
That properly to understand and explain living things, everything is relevant. I closely follow Noble on this point. He has what he calls the principle of biological relativity.
Yeah.
Where there's no privileged frame of reference, so to speak. You don't give DNA a special role. It has an undeniably significant role, but all of the levels, even up into ecosystem, are important. And that isolating one part, let's be honest, and I'll pick on Dawkins briefly here. Why does he focus on DNA? Well, part of it has to do with neo-Darwinian theory, but a big part of it has to do with you can't start with whole organisms. You can't start the evolutionary narrative with whole organisms. You have to start with some part of them, what he calls the original replicator, and accrete complexity around that as the story goes forward. So he is driven not really by the biological evidence, but by the naturalistic narrative he wants to tell, that once upon a time, there was the original replicator, the ribozyme that can make copies of itself. It was encapsulated in a lipid membrane. Darwinian selection began, and we run the evolutionary narrative forward. The reason to believe that is not evidential. The reason to believe that is that's a reductionist, materialist narrative that he wants to tell.
Yeah.
And I'll tell you what would be interesting, and I hope he's with us for another decade at least, is to watch Dennis Noble himself and the other members of this, call it this third way community, as they react to the growing number of intelligent design theorists. So this past weekend, I was with a very talented group of younger people from all over the world who are enthusiastic about saying, let's take off the mantle of naturalism, recover the explanatory tools that the pre-Darwinians had, like Cuvier, Linnaeus, and others, and approach the evidence with all the possibilities at our disposal. And I would love to see how Dennis Noble reacts to that, because it takes courage to say, you know what, I'm not just going to depart from neo-Darwinian theory. I'm going to depart from a naturalistic framework also. So systems biology, to me, is on let's take the sort of a planetary model of the sun, Mercury, Venus, Mars, and so forth. In relative distances, Mars and Earth are really close compared, let's say, to Earth and Jupiter.
Yeah.
or Earth and Neptune. So on that planetary model, systems biology and the Earth sit right like this, right? And you can practically walk from one to the other. And Subor, it's such an exciting time.
Yeah.
We have biology's golden age in front of us. Physics can't build accelerators big enough to test its theories. And we have more data than we know what to do with.
Absolutely. Thank you so much for giving your thoughts. I'm sure a lot of people appreciated that. And a lot of people don't know the significance of this debate. So the way that you've put it, I'm sure they will now go back and actually watch the discussion and realize what a landmark event it actually was.
Well, I will send you the references, and I'm going to check a few things that I'm worried, like the title of that paper were slightly inaccurate, and I'll try to clean that up. So I'm glad to have the opportunity to correct my own errors in real time. So it's great to see you again, and have a good week. And
Thank you so much for your time.
All right. Take care.
Cheers. Thank you, Paul.